The title of our presentation is Successful Generation of Human Muscle Fibers in Host Mice. We are Amy, Olivia, Layla, and Angela, and our advisor is Dr. Joseph Roche. We're students in the DPT program at Wayne State University, and this project was performed as part of our research curriculum. Skeletal muscle is capable of regeneration because it has stem cells known as satellite cells. When muscle fibers are damaged excessively, these satellite cells get activated. They divide to form numerous daughter cells, and then the daughter cells fuse to form new muscle fibers. These newly formed muscle fibers can replace muscle fibers that are severely damaged and undergo cell death.
In disease conditions like muscular dystrophy, muscle regeneration reduces over time, and this causes muscle wasting. In this slide, we compare a healthy muscle to a dystrophic muscle. It is evident that the healthy muscle is much larger than the dystrophic muscle. The dystrophic muscle is smaller and has undergone wasting because the regenerative potential has decreased over time. In order to replenish and rejuvenate regenerative potential, the Roche Lab at Wayne State University has developed a technique known as minimally invasive muscle embedding, or MIME for short. Through MIME, a piece of donor tissue is implanted into a host muscle, and the donor muscle tissue, along with its satellite cells, helps produce donor-derived muscle regeneration in the host muscle. We used the MIME technique to implant cadaveric donor muscle into immunodeficient host mice. The host mice also expressed a green fluorescent protein, which helps differentiate host from donor muscle fibers.
The results of our study indicate that at 12 weeks after performing MIME, we were able to detect human muscle fibers in a host tibialis anterior mouse muscle. In this picture, we are showing a cluster of human muscle fibers identified by the expression of a human-specific mitochondrial marker. This marker not only helps identify human muscle fibers, but it also confirms that the human mitochondria present in these fibers are in a healthy state, which means that the muscle fibers that have them are alive. Our results also indicate that animals that underwent the MIME procedure recovered contractile function just as well as the animals that underwent a sham procedure. These findings suggest that the human muscle fibers that are present within the mouse are likely contributing to force production. In addition, our data confirms that the cluster of fibers identified by the human-specific mitochondrial marker also expressed human-specific nuclear markers as well as human-specific plasma membrane protein markers, like spectrin.
There are several limitations in our study. One of them is the fact that this is at a basic and preclinical level. In order for these findings to translate into clinical treatments, clinical trials will have to first be performed. If cadaveric human tissue is transplanted into a living host, there is a concern of host rejection, and therefore lifelong immunosuppression will be needed. To overcome this limitation, the Roche Lab is working on developing engineered muscle tissue that can be generated from the host's own cells, which are known as autologous cells. We want to thank our advisor, Dr. Joseph Roche, Moriam Begum, who is a research assistant in his laboratory, and we also thank the various funding agencies, including Wayne State University, the National Institute of Health, as well as the Alliance for Regenerative Rehabilitation Research and Training. Thank you so much for your attention.